All right, so in the past modules, we've discussed Sanger sequencing, next generation sequencing, and now we're going to zoom in on a particular aspect of next generation sequencing called exome sequencing. So one of the ways that researchers can get more bang for their buck in sequencing is by focusing on just the portions of the genome that they're most interested in. And in medical genetic studies, for the most part, we're most interested in the protein coding areas, or the exome, the exonic regions of the genome. So this is only about, around 2% of the whole genome, depending on your particular estimation. And it has most of what the sort of well-characterized functional regions are. So rather than spending your money getting coverage of the entire genome, you can instead focus on just this 2% that has sort of high a priori expectation of having a functional consequence, and do a lot deeper sequencing, so you can be a lot more confident about the bases you see at these areas, rather than spending money sequencing portions that you are not as interested in. So I like to think of the exome as kind of the slice of the apple. So if you imagine this genic region of DNA, we have our yellow portions, which are our exons, with these intervening white introns. Of course, there's intergenic regions flanking on either side that aren't shown. The exome is just targeting these coding areas and picking out the couple percent of the genome that we're interested in this way. So in exome sequencing, you effectively design probes to target just the start of these exonic regions of interest. So in general, you'll capture on the order of 25 to 60 megabases of sequence, and because of this more limited amount of sequence, you can get to much higher depth for the same price. So we're going to go through how exome sequencing actually works in the next few slides. But the main thing to consider ahead of time is that it relies that you know where the exons are. So you already have your reference genome annotated for what exons you're trying to target. All right, so we start off in a similar way to how we did with the other sequencing technologies we've discussed. You fragment up your DNA into these short bits. And now we're only interested in the colored fragments that are going to be coming from our exons of interest. And these are the only things that are actually going to even hybridize to our probes. So the primary difference in whole exome sequencing is that our probes now, which can be either in a kind of standard microarray format that we talked about previously, or in solution on beads, but they work the same way. It's just fixing your DNA fragments to a physical location. But now these oligos or probes are actually targeting your sequence itself. So rather than a primer or some sort of linker that's actually going to be what's fixing your strands to a physical location, now they're targeting your known DNA sequence. So the fragments themselves are going to bind or anneal to your probes on the array and get fixed into that particular position. And then sequencing is going to proceed in basically the same manner as we talked about for next generation sequencing. So one major consideration for whole exome sequencing is that it really matters what you're considering to be the reference genome. So your fragments might bind better or worse to your probes depending on how well the predicted DNA sequence actually matches your reference genome. Here now, I'm showing an IGV screenshot of an exome collection of reads. So if you remember before from the whole genome sequencing reads, this coverage plot showed sort of a solid line across the same level of coverage over the entire genome. Now we're seeing these peaks in coverage around the exons. So I have this gene schematic at the bottom here showing where the exons are located, and you can sort of follow them up and see that most of these clusters of reads are mapping to these exonic positions. You can also notice that there are some spots on the exome, for example, here, where there's a decreased amount of binding. So even though this is in an exon, we're not seeing very many reads actually coming from this location. This indicates that our sort of target probe for this area didn't do a great job at binding DNA there. So probes can be more or less effective at actually capturing the DNA, and this has a really big impact on your downstream genotype calling. So one major consideration for whole exome sequencing is that it really matters what you're considering to be the reference genome. So your fragments might bind better or worse to your probes depending on how well the predicted DNA sequence actually matches your reference genome. Now I'm showing an IGV screenshot once again, this time for a collection of exome sequencing reads rather than whole genome sequencing reads. So once again, we have our chromosome at the top showing where exactly in the genome we're talking about or focusing in on. We again have our coverage line showing how many reads are covering every particular base. And instead of having a solid sort of block of identical coverage across this whole window, as we saw in the whole genome sequencing example, we have these peaks in sequencing focusing around particular regions, which, if we look at our gene schematic at the bottom of the slide here, correspond to the location of exons. So once again, we're aligning our fragments, now shown in these two windows here. So we're looking at two different individuals and the reads for each of them. We can see that we're mostly covering the exons, but there are a couple places in there that are just not getting as good coverage. For example, there's this sort of gap in coverage here, which even though we can assume that there were probes targeting this area, they're just not doing as good a job at binding the DNA or getting good read coverage in this particular region. And this is really important to think about for downstream filtering. If you get a region in an exon that's not got good coverage, you can't be as confident about the DNA calls or the genotype calls at that position. So to get even more bang for your buck, you can focus only on a particular panel of genes. So instead of sequencing the entire exome, you can sequence just the coding areas, for example, of genes that have been previously implicated in a particular disease or phenotype. So this is another way where you can limit the overall amount of sequencing in terms of regions that you have to sequence and focus instead on getting really deep coverage, really high accuracy of a few key areas that you think are really functionally important. The sort of overall goal when you're designing your experiment and choosing which type of DNA sequencing technology you would like to use is to balance the cost, how much money you have to spend on the project, versus how deep you need to have your sequences get, how confident you need to be in your genotype calls. And that depends on the end goal of the particular study. So for whole genome sequencing projects, as I mentioned before, you usually get on the order of 20 to 40 X coverage, or 20 to 40 reads covering any given base. You can get deeper than this in whole exome sequencing, so around 80 or more, simply because you don't have to spend your money elsewhere, as we discussed. If you wanted to do some sort of targeting sequencing study, so again, focusing on just a panel of genes related to a particular phenotype of interest, you can get several hundred X deep and be very, very confident from a medical perspective. Another strategy for getting, instead of a sort of exome-only sense of DNA sequence variation, is to focus on SNPs. So in this way, you can assess sort of a genome-wide pattern of variation, but again, reduce the number of sites that you actually have to genotype. And in that way, save a lot more sort of funds while getting a global perspective on variation. So in this module, we talked about how whole exome sequencing works, what distinguishes it a bit in terms of its technical methodology from your standard whole genome sequencing pipeline, and also why you can get deeper coverage for a cheaper cost compared to whole genome sequencing projects. So with that, I'm going to transition from talking about exome sequencing to talking about SNP arrays and how SNP genotyping works.